Hi guys, so today I'm going to be talking about G protein coupled receptors, or GPCR, in oil pastels. I just thought they were fun, they were colorful, they were sitting in front of me on my desk, so I think that this will be kind of a cute little way to show how G protein coupled receptors work. So here's my cell, and so first things first, so as we all know, cells maintain an environment that is distinct and discrete from their external environment. And the lipid bilayer is only semi-permeable, and it does not allow large water-soluble compounds, for example, water-soluble hormones, to pass through the membrane. G protein coupled receptors are the solution to that, and they allow these water-soluble hormones, which are my little blue dot, to send their message through the membrane without having to actually pass through it. So that green squiggle is my receptor, and the pink dot is supposed to demonstrate the G protein that is coupled to the receptor, hence G protein coupled receptor, the name. This purple box is the effector protein, which you will see the pink dot, the G protein. Once the water-soluble hormone binds to the protein receptor, it causes the G protein to be activated and to move over and send its signal to the effector protein, this purple box. The effector protein then, in turn, uses ATP, this little yellow star, because it's full of energy, to activate cyclic AMP, or it might cause it to activate a different secondary messenger, but cyclic AMP is a common one, so that's what I chose to use here. This secondary messenger, whether it's cyclic AMP or something else, will then activate a protein kinase, which is an activating enzyme that can cause a lot of different cascades. They have many functions depending on the protein kinase. And I apologize for this weird little blob. I was running out of oil pastel colors, so I labeled it protein kinase. This protein kinase then causes a cascade of events that produces the desired result that the hormone originally sent the message for the cell to undergo. So I know it's a bad angle, sorry, I have limited space to work with, but here is it all encompassed. Again, the blue dot, the water-soluble hormone that cannot pass the cell. The green squiggle is my G coupled protein receptor. The pink dot is the actual G protein. The purple square is the effector protein that the G protein signals. The effector protein, the purple box, then uses the yellow star, which is ATP, to activate a secondary messenger. The secondary messenger then in turn activates protein kinase, which causes the rest of the cascade that then regulates or activates or inhibits different cellular activities. Okay, so my voice is a little bit out of sync in the last section, so I apologize for that. Ended up taking pictures of the rest of what I was trying to show so that I could keep it more consistent. But basically, I just wanted to go into more detail about the components of the G protein coupled receptor. So the first component that I'm going to go into in more detail is the G protein itself, which is, as a reminder, is the pink dot that the arrow is pointing to. So G proteins have three subunits. They have an alpha, a beta, and a gamma subunit. And the alpha subunit in particular is special because it comes in multiple forms. So in class, we focused on the alpha S and the alpha Q forms of the alpha subunit of the G protein, so those are what I'm going to focus on in this video. The blue dot next to the alpha S and the purple dot next to the alpha Q are not meant to be related to the picture of the cell. They are color coordinated for the rest of the information that I am going to give you that pertains to these two subunits. So that's why alpha S is blue, alpha Q is designated by purple. So what makes these different forms of the alpha subunit unique is that they have their own different effector proteins. So the different alpha subunits have different effector proteins that they activate, and the different effector proteins are in turn what cause such unique and what help to cause such unique and specified cell reactions to water-soluble hormones. So the effector protein for the alpha S subunit, again color-coded in blue, is adenylyl cyclase, and the effector protein for alpha Q subunits is phospholipase C. So these effector proteins, in turn, activate different secondary messengers. Adenylyl cyclase activates cyclic AMP as its secondary messenger, and it does so using an ATP. Phospholipase C, by contrast, actually cleaves a phospholipid from the cell membrane into IP3 and DAG. Those are its secondary messengers. And these cause calcium to be liberated within the cell and activate protein kinase C. So this is just a final picture of my entire G protein coupled receptor in oil pastel. I hope that this was a good review of all the things going on, and I hope you enjoyed all of the bright colors. Thanks.